And so then you add this, and it goes on, for a little bit longer. Okay, so you've got this nice strong labeling that's occurring. And then you're, moving to the chase phase, where you're throwing a bunch of thymidine without sunflowers. Okay, so there's, this causes a dilution. So now the amount of radioactivity that's being incorporated starts to become diluted, and you can see it evident in these regions right here. So, the further and further away from where the replication started, you get this dilution that's occurring. So there's a little bit less labeling, less and less. And so the idea is that if you put this in, this pulse chase experiment, you're looking at something that is undergoing replication, you should definitely see a gradient of labeling, because the DNA that's actually being synthesized here, right here, where there's a lot of tritium, you'll get incorporation. But when it's synthesized out here, the amount of tritium is kind of decreasing, so you'd expect this kind of gradient. And the other sort of trick that goes with this is that if you're looking at these fibers spread on a microscope slide, so if you have a piece of replicating DNA, you sort of have this, replication bubble, as illustrated here. But when you stretch the fiber onto of the DNA, you're basically pulling the ends apart, or pulling on the ends so that it collapses into a line. So you get these long fibers. And so the anticipated result is shown over here on the right. And again, it's a very bad illustration, but this is where you sort of have a very heavy labeling initially. So we would see it here during, the pulse phase. So there's lots of labeling. And then once you introduce the chase, which would be occurring here, the labeling becomes less and less still. Okay, so it's not as intense. So you kind of go from this very heavy amount of labeling to something that looks like that towards the end of the fiber. So all we're looking for here is distinguish unidirectional from bidirectional. So if the DNA is being replicated bidirectionally, you see this dilution on both sides, as represented in this figure, of the newly synthesized region of DNA. Below, if it's unidirectional, then you should only see this dilution on one side. So it's relatively clear distinction between whether it's bidirectional or unidirectional. All right, so what do the results look like? So on this slide, I have an audio radiogram, and I think it takes a little bit of a careful eye to actually look at it. But once you sort of start looking at things, like obviously all these dark things here, so this one here, this one here, here, these are all replicated regions of the Chinese hamster ovarian cells DNA. And then you can ask, do you see this dilution effect? And I think if you look at it carefully, here's a very nice example. Okay, you can see that it's quite heavy in the middle, so it's very dark. And then it kind of peters off on the ends. So over here it's very light, and again over here it's very light. Okay, so as it peters off, there's less intense labeling on both sides of it, so that would be consistent with bidirectional. So this is a pretty good example where it's heavy in the middle, and then it gets quite a bit lighter at the ends of at each of the ends. Again, this one similarly over here, heavy in the middle, and then it gets lighter and lighter at the ends as well. So pretty much, everything that they examined, of course, they went through a bunch of these images. They quantified the fraction, and they showed that this bidirectionality versus unidirectionality pattern, and primarily or predominantly the pattern that they saw was consistent with bidirectional replication. So this then was the evidence that eukaryotic chromosomes are replicated bidirectionally, and it was actually one of those sort of rare instances where they figured out in a eukaryotic cell first, and then other groups went back and actually did it in E. coli and came to a similar conclusion. The other thing that I want to point out on this is sort of interesting, and it was actually the first visualization of replicating eukaryotic chromosomes. So again, it was the first visualization of replication. And one thing that I want to point out in this picture is that there's actually one fiber that kind of goes like this. So if you follow it all the way through, so that's one piece of DNA. And what's cool about this is that you can see this one piece of DNA has multiple positions where replication is initiating. So we have one position here, one here, again another one here, here, and down here. So on this one fiber, there's 4 different regions. 1, 2, 3, 4, 5. There's 5 different regions. Different regions where replication is starting. So we have, 5 different regions where replication is starting. And so this is obviously, quite different than E. coli, because for the E. coli chromosome, there's only one replication origin, so it's different. From E. coli, where there is only one origin of replication. Okay, so basically this opened up, just a ton of different questions about how replication is controlled on eukaryotic chromosomes. Like how does it know where to start here instead of over here? So how do you start, over here and then you start over here again? How do you know where? Which, are obviously physical separate places in space and at different times. So it is from a very simple experiment. We've introduced a lot of sort of interesting mysteries that were then followed up in quite a bit of detail. And we'll delve into that later on in this lecture. Okay, so that's bidirectionality. All right, so in this slide at the top, that's Joel Huberman with kind of a decent, almost neck beard. And then I don't know, this is like Art Riggs below, looking extremely 1970s. If you take into account, the mustache, the glasses, and the unbelievable hair. Okay, so just another perspective on those scientists there. All right, so now we'll delve into how replication is semi-discontinuous. So the conundrum here is that we know from Messelson and Stahl that replication is happening in a semi-conservative fashion. So we're separating the 2 DNA strands and using each of them as a template for new synthesis. We also know from Watson and Crick that one of these strands goes 5' to 3', and the other one goes 3' to 5'. So this raised the question as to if the strands are being separated, as in Michelson and Stahl, and they are replicated at a fork like Huberman and Riggs. The two strands are going in different directions, and we also take into account that Kornberg said the polymerase only goes 5' to 3'. How are we dealing with all of this? So again, we have the one template being in the right orientation, and then the other template being in the wrong orientation. So what the Okazaki set out to do was, again, design an experiment that would distinguish between different models that could accommodate how this synthesis was happening. And on the slide, we have those models illustrated. So the first model, Model A, which we'll focus on here, is that synthesis could somehow be continuous on both strands. This is from their paper. I'll just make one adjustment here. That we're going to the 3' end over here, and this is the 5'. Okay. And again, the arrow should be this way, not as depicted here. Okay, so just make that correction. But nonetheless, this prediction was somehow that both strands would be separated and would be replicated continuously. So maybe they are just totally unwound so that they are, completely available for synthesis, and then they would just go in opposite directions. Another possibility was this Model B, which was the semi-discontinuous model. So this involved, one strand being replicated 5' to 3'. And as you know, on the template, that is being represented in the right orientation. So this would have been continuous. And then in contrast, the second strand is replicated in pieces, again 5' to 3', but discontinuously. And that's why it's termed semi-discontinuous. So this would be replicated in pieces. So just to reiterate, a semi-discontinuous, you have one strand that's continuous and one strand that's discontinuous. Model C suggests it's discontinuous on both strands. So even though they're presented in different orientations, both of them could be synthesized in pieces. So both of them discontinuously. And then finally, there's this last model, Model D, which I think sort of derives from the random dispersive thing that we saw with Michelson-Stahl, that, maybe there's some really nutty backbone cleavage that's going on, and the whole thing is just synthesized as a jumble of pieces. So what they sought to do was distinguish or, identify between these different models, which one was at play. And what they reasoned was, so, if you synthesize DNA in short pieces, is it going to stay in short pieces? I mean, is your genome, if you really think about it, made up of a whole bunch of little short pieces? So no, right? I mean, it's made up of entirely double-stranded DNA. So even if it's synthesized with short pieces, those short pieces must rapidly be joined together to give intact double-stranded DNA. So the thinking that these would be rapidly joined together, they thought what they really needed was a method where they could capture the very, very most brand newly synthesized nascent DNA. Okay, so they needed a method to capture newly synthesized DNA. Okay, so let's talk about the experiment. And what they did again is kind of the pulse labeling strategy. So they too, labeled their cells with radioactive thymidine, so tritiated thymidine, for short amounts of time, so that they were very, very serious with regarding these short periods of time, because when they applied their label, they wanted to be able to capture these tiny pieces of DNA before they got joined together. Okay, and so they would stop the reaction by, taking the culture and pouring it onto ice. And this ice contained cyanide. So after short periods of time, they would stop reaction by pouring onto ice with cyanide to kill the cells. Okay. So, hopefully after this short period of labeling, they could capture these small pieces when they're synthesized and then just kill everything dead. So basically they would then separate on the basis of size, right? Because what the goal of this experiment is that they're looking for small pieces. The prediction, that is, in most of the models, except for the entirely continuous one, is that there will be some small pieces of DNA. So, they want to separate by size, and they do this again by a gradient and ultracentrifugation, but this time it's a little bit different. They're going to use an alkaline sucrose gradient, and so the idea with an alkaline gradient is that the alkali denatures the DNA. So denatures DNA. Okay, and so why does this alkaline gradient, which really has a high pH, so why does it denature a DNA? Can you think? In all honesty, it really has to do with the hydrogen bonding, because at the high pH you get protonation of the bases, and deprotonation of the bases, and that would disrupt the hydrogen bonding. So, at any rate, the goal here is to separate the two strands. And the reason that this becomes important is actually illustrated down here. If you don't separate the DNA strands, you have your DNA that's being made at this, this replication fork, right? And everything is hybridized together, so this is going to migrate like one very high molecular weight species, even though we're interested in these, these tiny nascent fragments, which are obviously of lower molecular weight. So if you allow this thing to still be annealed to the parental template, we're not going to be able to discern its actual true molecular weight. However, if we, denature the DNA at a high pH, now the constituents of the strands are separated from each other. And this thing over here will migrate at a higher molecular weight than this thing that's down here that will have a lower molecular weight. Whereas the parental strand will totally be as the most highest molecular weight. Okay, so, the strategy is kind of the same. We have, our sucrose gradient. So I'll just draw this here. So this is our sucrose gradient. Okay, and in this case we layer our DNA extract on the top. So we add in our DNA. And then we do the centrifugation, and then, the DNA migrates in the gradient according to size. Okay, and what happens is we'll have the small fragments at the top and the larger at the bottom. Okay, because the DNA migrates according to size. So this is a little bit different than the cesium gradient. We're not looking for it to stop at the position where its density is equal to the density of the gradient. We're just kind of letting it go on the basis of size. So small stuff doesn't migrate in that well. Large stuff migrates in better. So again, the small are at the top, and then the larger are near the bottom. So then how are we detecting the DNA? So we're using tritiated thymidine. So what we need to do is actually fractionate this gradient. So we can undergo fractionation. And the way you fractionate this gradient, you just take a needle and you poke a small hole at the bottom of the tube, and you let the drops fall out. And then you just collect, some of the tubes, some of fractions. Okay, and so the first tube will actually have the large stuff, and then the last tube will have the small stuff as it migrates through the test tube. So the small stuff. Okay, and then you just count the radioactivity, and then you put it on a graph, which is what we actually see here. Okay, so now let's focus on the data and on the graph that's on this slide, and talk about the important details. So you remember they did this experiment several different time points with either 2 seconds of labeling, 7 seconds, 15 seconds, and so on. And those are highlighted on the graph. And let's just focus on the 7-second line because it's a very short labeling time point. And what I hope you can see is there's actually 2 peaks, right? There's this one here. So there's a peak at the top of the gradient, and that's outlined here by the word top. And then there's this short, sort of broader peak stretching towards the bottom, right? So this is the bottom of the gradient. So what's the stuff in the kind of sharp peak near the top? It's the small stuff, right? Because we're at the top of the gradient. And then, the stuff near the bottom, of course, is the larger stuff that's being synthesized. Okay, so what they infer from this is that it appears that the DNA is being synthesized, even at early time points, as 2 different species. So one short and one large. And this theme continues, like if you go out to 15 seconds, we see the same type of, time point graph. And then again, even at 30 seconds, we have a nice peak with some smaller material, and then, the larger material. And you can actually see that the center of this larger material is actually kind of like shifting to the right, okay, at each time point. So even though this kind of looks sort of like it's continuous synthesis, the strands are actually being elongated and getting longer. But the idea or the inference from this data was that at the early time points, the DNA is about half and half in small pieces and half in large pieces. So if you refer back to the models that they were trying to distinguish between, this really is most consistent with the semi-discontinuous model, right? Where one strand is synthesized continuously as large pieces, and then the other is synthesized discontinuously as small pieces. And of course, the small pieces we now call Okazaki fragments. So, if you do an experiment carefully, you can get a small piece of DNA named after you. So that's pretty exciting. So one takeaway thing from this data is that the DNA is being synthesized as both large and small fragments. And small fragments. And that model is actually very consistent with the semi-discontinuous model. And they name the small fragments, or the small pieces, Okazaki fragments. Okay, so one last thing with this data is, again, if you go to the longer time points, so say we go out to 60 seconds or 120, what happens? Is it still half small, half big? No, actually, it looks like the proportion that is large is actually increasing, and this is consistent with their expectation, right? Their whole basis for the design of their experiment was that these very short pulse label time points was that they would expect these, small fragments would be rapidly joined together into big fragments, and that's what we're actually seeing at these later time points. So the proportion of big fragments increases, presumably because these small fragments that were being generated down here become joined together, although there's still some small amount of them being synthesized. Yeah, so these older ones are being joined together into larger pieces of DNA. Okay, so that's another takeaway from this. At longer time points, the proportion of large is increasing. And that's the expectation that the small pieces are being joined together. So that you get more large pieces. Okay, so that's the semi-discontinuous model. And here are the Okazakis, very charming couple, beautiful laboratory. So it's always good to be able to put a picture to a name again. All right, so let's just take a step back here and just sort of briefly summarize what we've covered up until this point. We've basically seen that DNA polymerase synthesizes DNA directed by a DNA template. It synthesizes in the 5' to 3' direction. It also requires a primer. I've shown you evidence that replication in cells is semi-conservative, bidirectional, and semi-discontinuous. And so what this sort of prompts you now to ask is the question about how this reaction can actually happen. Is this a reaction that could be carried out by a single enzyme, namely DNA polymerase? Or if you sort of consider what's involved in the reaction as a whole, I think it's pretty easy to come to the conclusion that DNA polymerase could not carry out this reaction on its own. And first and foremost, DNA polymerase can't unwind double-stranded DNA to produce a single-stranded DNA, and the template of, as you know, for DNA polymerase is, of course, single-stranded DNA annealed to a primer. So the question becomes, how do you get from, this double-stranded piece of DNA to a piece of primed single-stranded DNA? So again, here's our primer with our 3' hydroxyl. Another thing is that single-stranded DNA is not particularly stable. So, what's going to happen to single-stranded DNA? whether in a cell or in a test tube. You have the double helix. You unwind it. Does the single-stranded DNA stay single-stranded? So that's the question. What could happen to it? What does happen to it? There's a couple of things that can happen. It can actually, reanneal, right? So the two strands could actually come back together. It's also possible, like a lot of single-stranded DNA could have elements that would provoke, the formation of secondary structures. So if there's repeated elements within the DNA, you maybe get weirdo hairpin things forming. So you can get secondary structures, which could look like something like this. Okay. Who's more susceptible to nucleolytic cleavage, single-stranded DNA or double-stranded DNA? So, single-stranded DNA is, right? So you get cleavage. So there's a lot of reasons why cells prefer not to have single-stranded DNA hanging around, yet single-stranded DNA is a prerequisite for the polymerization reaction. So the question is, how does that happen? Point number 3, DNA polymerase can't start DNA chains de novo, right? So it needs a primer from somewhere. So again, let's just highlight that, needs a primer. How does this get made? Who makes it? Okay, how does this even happen? Even polymerase 3, which we're all convinced is the polymerase that replicates the E. coli chromosome, is a replicated polymerase that is very slow and too distributive. So again, I can give you an example of the E. coli's genome. So again, here's the point that DNA poly 3 is too slow and too distributive. So again, if we write E. coli's genome, there's 5 million base pairs. The rate of synthesis for poly 3 in vitro is approximately 200 nucleotides per second. And this thing is really cranking, so that means you need 25,000 seconds, and you divide this by 2 because there's 2 replication forks. Okay, so that's 12,500 seconds for poly 3 to replicate the E. coli chromosome. So that's pretty great, right? So that's approximately 200 minutes. But sadly enough, E. coli doubles in about 20 minutes. So, but E. coli doubles in 20 minutes. So there's a bit of a gap here, right? So if we go from 200 to 20 minutes, so what contributes to making the polymerase more efficient and more processive in cells? And then, of course, there's that issue that the polymerase only goes in one direction. So how are the reactions on the leading and the lagging strand coordinated with each other? All right, so let's not keep you in suspense any longer. I know you're all dying to hear how DNA gets unwound. So here it is on this slide. The DNA helicase, basically the enzyme that unwinds the DNA is called DNA helicase. And what I wanted to show you first off is a very simple assay for DNA helicase activity. And then I'll show you some of the properties of the DNA helicases. So the assay is as follows. What we're looking for is an enzyme that can convert double-stranded DNA to single-stranded DNA, right? So, presumably the substrate we're going to give it is double-stranded DNA. So if we give it that, we're going to hope it makes some single-stranded DNA. So then we need to be able to distinguish between the double-stranded DNA and the single-stranded DNA. And I'm sure you can imagine that one way to do this would be on the basis of size, right? Because, a double-stranded fragment of X nucleotides in length is going to be twice the molecular weight of a single-stranded fragment of the same X number of nucleotides. So the assay really is like this. You make 2 pieces of DNA. And these are typically synthetic DNAs, so oligonucleotides. You label one of them, and again it's depicted up here, with P32 so that you can follow it through the reaction. And the way these DNAs are designed is that they have a region of complementarity. Okay, and so it's the double-stranded DNA that we're looking to unwind. And then typically you'll have these single-stranded tail end regions, right? And the reasons for the tails is that it was presumed that if you just presented an enzyme with a blunt-ended piece of double-stranded DNA, it probably wouldn't know how to sort of invade the double-stranded DNA to produce single-stranded DNA. So the thinking was maybe if we just give it some single-stranded region to kind of latch onto, that would help the reaction. And so it turns out that it typically does. And typically, these reactions are done in the presence of ATP. Okay, so we add in some ATP because you might imagine that separating the 2 DNA strands is going to be a process that's going to require some energy. So an ATP requirement would also be reasonable to infer. And at the end of the reaction, the prediction is that you have this double-stranded, template that was up here becomes unwound, and you have 2 single-stranded moieties. One of which, of course, is labeled with the P32. So now the game is just to separate the double-stranded from the single-stranded, and this is typically done by gel electrophoresis. So what we see over here, this would probably be an agarose gel. Okay, maybe an acrylamide, who knows. And the direction of migration would be as the arrow presents, so from moving downward through the gel. And the idea is that double-stranded DNA is going to migrate slower through the gel than the single-stranded product DNA. And then we can identify who's where by virtue of the P32 radioactivity. Okay, so for any experiment, you really need to have some positive and negative controls, right? So I'm going to pose the question, what would be a reasonable negative control for this reaction? I mean, we really want to convince ourselves that if we see separation of the DNA strands, it's because we've added this DNA helicase or a protein fraction, and not just because, the strands fell apart for whatever reason. So what is the negative control? Think about it for a minute. So really, the negative control would be, all of the components of the assay minus the helicase, so no helicase. Okay, and that's what's shown in lane 2, okay, in this lane right here. And as predicted, almost everything is in the double-stranded form, so this is what the double-stranded form would migrate at, and down here would be the single-stranded form. Okay. Also, there's sort of a positive control here, and I sort of say sort of because it's not an enzymatic positive control, but it's just to convince ourselves that if you denature the DNA substrate that you're putting in, one, you can tell the difference in size, and two, it's not going to just snap right back onto itself to form the double-stranded input. And that again is shown in lane 1. And again, there's no helicase here. So no helicase. And it's simply done by boiling. So applying some heat to denature the double-stranded into single-stranded components. So if you heat the substrate to denature it, the result is the strands remain denatured during the course of the experiment, and you can tell the difference between the size of the double-stranded and the single-stranded that's depicted here as these bands. Okay, so now we're ready to go. And what they're adding over here from lanes 7 to 10 is the DNA helicase called MCM. Okay, and this is the DNA helicase in eukaryotes like ourselves. And what they've done is they've added increasing amounts of the MCM complex. Okay, so increasing amounts as we go through lanes 7 to 10. So this is the DNA helicase. Okay, and obviously this is a very common strategy in enzymatic reactions, that you like to see some kind of dose response, that if you're putting more of the enzyme in, you expect to see more activity. And so that's what they're actually doing here. And indeed, this is what they see as you add increasing amounts of the MCM. You clearly see the production of more single-stranded DNA. And obviously the amount is more here than it is over here, lower concentrations. So pretty nice assay, pretty simple. And this is essentially what was used to originally detect DNA helicase activity, and then subsequently used for different kinds of characterization of the many different helicases. This slide actually shows some of the helicases, what they look like. So basically, as illustrated here, all of the helicases are composed of identical monomers. So we got a couple of them here. One is from a bacteriophage, and this one here. One is from a monkey virus. This one is from archaebacteria. And again, this one is from E. coli. So what do you notice about these helicases? What do they all have? Well, they all have 6 subunits, so they're all hexamers. Okay. What else do they have? They have a hole, right? So they're all like these donut-like structures. They are toroids. You might imagine that this hole does serve a purpose, and it does. The DNA can actually pass through this hole, so we can have our DNA through here. And I forgot to mention that all these hexamers are identical, so make a note of that. And they all have this central cavity. Okay. But the whole, however, doesn't really tell you all there is to know about these enzymes, though. You might be curious as to whether a double-stranded or single-stranded DNA passes through this hole. And unfortunately, it's big enough to accommodate both. So both double-stranded and single-stranded DNA are accommodated. So, typically, the size of this hole doesn't really help us in that regard. The hole does have some characteristic charge properties, and I'll pose the question, what charge would you expect the residues lining this donut hole to be comprised of? And hopefully you've guessed correctly. They would be positive charges that are lining the cavity, because DNA is negative, so this obviously would make sense that it would be positively charged. The replicative helicases are found obviously throughout evolution, and they're quite remarkably conserved in terms of what they look like, as we can see from these various species on the slide. They're not quite as conserved as in terms of their mode of action, but as you might imagine, as you move to more complex organisms like ourselves, the story tends to get a little bit more complicated. All right, so here is the MCM complex from eukaryotic cells, so what we were just talking about a few minutes ago. So this is the DNA helicase that unwinds DNA during DNA replication in all eukaryotes. It is, as the other enzymes I showed you, composed of 6 subunits. But in this case, 6 subunits are distinct from each other. So there are 6 different proteins that make up this hexamer. So it's unlike in the cases that I showed you previously, where the hexamers were composed of identical monomers. So this thing down here, the MCM, is a heterohexamer. Okay, so a heterohexamer, 6 distinct subunits. The name MCM just means mini chromosome maintenance, and the reason they called it that is that they were all isolated from a particular genetic screen, the details of which I won't go into. So at any rate, the MCM complex, again, is a hexamer and also has, that central cavity in the middle that the other ones we showed in the last few slides. And then if you take this donut and you actually just turn it on its side, which is what is shown here, it's actually pretty sophisticated donut. It has a couple of lobes, so you can see them quite clearly here, sort of this, this N-terminal lobe and this C-terminal lobe down here. And then it also has the channel. The channel, and that would be running through the center of this thing. So there's a few odds and ends and different bits and pieces that I've shown you here. Up here is actually a schematic of the domain structure of the helicase. So each of these subunits has the same domain structure. All I want to point out here is, or take away from this, is that it has a domain that is responsible for, ATP hydrolysis. ATP hydrolysis. Okay, so each subunit has these domains. Okay, and so this, this ATPase domain, which is demonstrated right here, you can also find it in the 3D structure. And this is what's actually shown here. So this is one monomer. Okay, and on this side, this is the inner surface lining the channel. Okay, so this is would be in, and over here would be the outside. Okay, so this is the inner surface lining channel. Okay. And what I want to point out here is that the ATPase functionality quite clearly lies in the C-terminal lobe, and the ATPase faces inwards, so it's actually facing inwards towards the cavity. So the ATPase domain faces inwards towards the cavity. Okay? And again, this ATPase domain is responsible for ATP hydrolysis. Okay, so there's a couple of other interesting features of this thing that are illustrated here, and the importance of some of these I think will become apparent as we get a little further into the lecture. But the way this is proposed to work is by passing a single strand of DNA, in the case of MCM. It's the template for the leading strand synthesis. So the 3' end of the leading strand goes through the channel. So this is the leading strand. Through channel. And then the 5' end actually passes outside of the molecule of the NCM complex. So that's what's actually shown here. And what's sort of interesting about that is that there's some regions on the outside that can bind single-stranded DNA. So this lagging strand passes outside, and there's regions for binding this strand. Okay, and this poly alpha here is actually the lagging strand polymerase. And we'll get into that in just a few more minutes. So again, the MCM protein, will associate with a couple of the DNA polymerases, which is also interesting because, as I mentioned, you don't really want to have single-stranded DNA hanging around, so it's unwound. The DNA is presented very rapidly to the polymerases, so the leading strand DNA polymerase is actually, situated right here. Okay, so it's very close to the channel. Okay, so that's poly epsilon, and again, we'll talk about that in a few minutes. And it's situated right where its substrate exits the, the MCM complex. And the lagging strand, again, up here, polymerase is also situated here, very close to its substrate, which sort of goes around the exterior of the complex. So just to sort of draw this schematically, imagine we had a replication fork. Okay, so I'll draw 5' to 3', and then 3' to 5'. And the MCM complex migrates on the 3' strand. So the directionality of the helicase, so if we put our MCM here, is described with respect to the strand that they're moving on. So the MCM is a 3' to 5' helicase. And it's named on the strand that it's moving on. Okay? Um, so all helicases unwind DNA, but they don't do it all exactly the same. And so an example of a sort of a contrasting case is DNA B, the DNA B protein. So DNA B helicase. It's a replication helicase in E. coli cells. It translocates on the opposite strand. So you would find it here. So it moves in the 5' to 3' direction. Okay, just opposite, in the opposite direction of the MCM. Okay, so that's the enzyme that unwinds the DNA. So now we're at a position where we've produced single-stranded DNA from double-stranded DNA. This now can serve as a template for DNA polymerase, but we have this problem that we discussed earlier regarding the instability of single-stranded DNA. So if you remember, we mentioned it can possibly re-anneal. It has the propensity to form secondary structures, and of course it's very susceptible to nucleolytic cleavage. So the bottom line is that we have to protect the single-stranded DNA. And the way that DNA in single-stranded form is protected intracellularly is by the action of proteins that are referred to SSBs. So SSB stands for single-stranded DNA binding protein. Okay, so that's depicted up here at the top. And so you might guess SSBs bind to single-stranded DNA, and the purpose of their binding is really to protect it from adverse effects. Okay, so protect from adverse effects. Okay, and what I've actually so protect it from adverse effects, and to keep it in a single-stranded form such that it can be presented in that form to the DNA polymerase. And what I've shown in this slide is actually the eukaryotic SSB, which is called RPA. Okay, so the eukaryotic SSB is called RPA, and that just stands for replication protein A. Protein A. Okay. And it was named because it was an early identified fraction when Tom Kelly's lab was fractionating eukaryotic extracts to identify different proteins that were required for replication in vitro. So this was kind of, the first one that they came across. So RPA is basically a heterotrimeric protein. So it's a heterotrimer. Okay, so it's made up of 3 subunits, and those are colored differently here. So this green one is 70 kilodaltons. So this one here, 70 kilodalton subunit. And they basically just name them by their molecular weight. So this was called RPA 70. The blue one is RPA 32. And this little red one is RPA 14. And what's interesting about this from a structural perspective is that the enzymes function as a complex to bind single-stranded DNA. So the thing that's depicted in black here, running along, so